Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 3, Human Variation, Growth, and Adaptation. In this module, we investigate how and why humans differ, biologically, developmentally, and adaptively. Let's revisit the long history of how people tried to make sense of human differences. Early scholars treated variation as something that could be boxed into racial categories, as if humanity could be divided neatly into a few fixed types. But as you'll see, the evidence simply doesn't support those old assumptions. Physical traits shift gradually across geography, not in discrete racial blocks, and much of what earlier scientists attributed to race was actually shaped by environment, history, and lived experience. This shift, from rigid categories to clinal population-based thinking, is one of the most important developments in biological anthropology. From there, we'll look at how humans grow. Think about this. Every person's body is a record of biological potential interacting with environmental reality. Nutrition, disease exposure, stress, and social conditions all leave measurable marks on growth patterns, from prenatal development through adulthood. When we talk about life history, we're really talking about the timing and energy trade-offs that shape our growth, reproduction, and aging. Finally, we'll explore adaptation. Now, this part is important. Humans don't adapt in just one way. We adapt genetically over generations, developmentally as we grow, physiologically in response to immediate conditions, and culturally through clothing, tools, and social behaviors. High-altitude lungs, heat tolerance, cold weather body proportions, these aren't random. They're responses to the environments our ancestors lived in. By the end of this module, you'll have a clearer sense of how biology, environment, and culture intertwine to produce the incredible variety we see in humankind today. In this section, we examine the history of the race concept and the scientific evidence showing why race is a social, not biological, category. First, let's revisit the world in which the early ideas of race were formed. When European scholars began traveling and writing about human differences during the Renaissance and into the Enlightenment, they were encountering people from regions they had never seen before. Without the tools of genetics or modern anthropology, they relied heavily on what they could observe with the naked eye. Skin color, hair texture, and facial features became convenient shortcuts for trying to classify humanity. It felt systematic to them, even scientific, but it was built on limited information and strong cultural assumptions. As naturalists tried to organize the living world, they applied the same mindset to humans. Think about this. They were using the same logic they used to sort plants and animals into species and varieties. One influential figure, Johann Friedrich Blumenbach, proposed 5 broad varieties of humans. His categories, based on skull shapes and visible traits, were meant to reflect natural groupings. But remember, he was working in the late 1700s, long before we understood anything about chromosomes, genes, or population-level variation. Still, these early classifications became deeply embedded in Western thought. They created tidy labels that many people took as biological truths. These early racial categories weren't just scientific curiosities. They shaped how scholars, governments, and societies interpreted human difference. Because the classifications were framed as biological, they were often used to justify inequality. When we look back at the 19th century, we see scientists measuring skulls, noses, and body proportions in an attempt to rank groups along imagined hierarchies. Today, these methods seem obviously flawed, but at the time they were presented as objective and authoritative. As we continue through this story, you might wonder why these early ideas held on for so long. One reason is that they were simple. They gave people an easy way to make sense of the enormous diversity of human traits. A second reason is that they aligned with social and political structures of the time. When a classification system reinforces existing power dynamics, it becomes very hard to dislodge, even when the science begins to contradict it. But the turning point came when anthropologists and biologists started collecting more rigorous data. As global exploration expanded and scientific methods improved, researchers realized that human traits didn't cluster neatly into the categories created by early scholars. Instead, traits like skin color or cranial shape changed gradually across geography. This pattern, what we now call clinal variation, didn't fit the idea of discrete racial groups. The more scientists looked, the more they saw complexity rather than sharp boundaries. So as we trace the history of the race concept, what we're really seeing is a shift from surface-level observations to evidence-based population science. And that shift sets the stage for understanding human variation in a far more accurate and meaningful way. Let's take a deeper look at how biological anthropology dismantled the old idea that race is a fixed biological division of humanity. First, let's revisit the assumptions early scholars were working with. For a long time, people believed that physical traits like head shape, skin color, or body proportions were stable markers of separate human races. These traits were treated almost like genetic signatures that defined and separated groups. But once researchers began collecting better data and questioning those assumptions, the picture changed dramatically. A major turning point came from the work of Franz Boas. He didn't set out trying to prove or disprove race. He simply asked a straightforward scientific question: are the physical traits used to classify people actually fixed? To answer this, he studied head and face measurements in immigrant families in the United States. What he found shocked the scientific community at the time. Children born and raised in the U.S. had different cranial measurements from their parents who were born in Europe. If head shape were a rigid racial trait, that kind of change within a single generation shouldn't happen. But it did. Boas showed that environment, nutrition, health, childhood conditions could influence traits previously assumed to be unchangeable. Think about what that means. If supposedly racial features can shift based on lived experience, then they aren't reliable biological markers of distinct human groups. Another major voice in debunking biological race was W.E.B. Du Bois. Working at a time when racial explanations dominated public discourse, Du Bois noticed something crucial. Differences in health, disease rates, and mortality weren't rooted in biology. They reflected social inequality, poverty, discrimination, access to care, not innate racial differences. His insights pushed the discussion beyond bodies and toward the environments in which people lived. You might wonder why this matters scientifically. It matters because it forces us to ask whether observed differences come from genetics or from context. Du Bois showed that context carries enormous weight. As genetics advanced in the 20th century, the evidence against biological race became even stronger. One of the landmark contributions came from geneticist Richard Lewontin. Lewontin examined genetic variation across a range of human populations and asked, how much of this variation actually aligns with traditional racial categories? The answer was surprising to many at the time. He found that most human genetic variation, about 85 to 90%, occurs within populations, not between them. Only a small fraction was associated with differences between the so-called races. In other words, two individuals from the same race can be as genetically different from each other as two individuals from different races. This finding made it very difficult to argue that races were deep biological divisions. The genetic data simply didn't support it. Let's pause for a moment and think about this. If traits change with environment, and if most genetic variation exists within groups rather than between them, then the old racial categories weren't describing biological reality. They were describing social perceptions. And once those perceptions were treated as biological facts, they became powerful and harmful ideas. Another key insight from modern anthropology is that human traits rarely cluster into neat, bounded packages. Instead, they shift gradually across geography. We call these patterns clines. Skin color, for example, becomes gradually lighter or darker depending on latitude and UV exposure. There is no line on a map where one race ends and another begins. The same is true for many other traits once used to define races. When we map the data, we see smooth gradients, not sharp boundaries. These clinal patterns reflect gene flow, migration, local adaptation, and the long interconnected history of human populations. So what does all of this tell us? It tells us that race, as a biological system, doesn't hold up under scientific scrutiny. The categories created in the 18th and 19th centuries were based on appearance, not on genetics or evolutionary history. As anthropology and genetics became more rigorous, those categories were revealed to be culturally constructed rather than biologically grounded. By understanding this scientific journey, from Boas's studies on cranial plasticity to Du Bois's observations about social inequality to Lewontin's genetic analyses, we see how the concept of race shifted from assumed biological truth to recognized social construct. And that shift is essential, not just for anthropology, but for how we understand human diversity today. Let's look more closely at the idea of clines. Think about standing on a long stretch of coastline. As you walk, the environment doesn't suddenly flip from one ecosystem to another. It changes step by step. Human biological traits often behave in exactly the same way. Skin pigmentation is the clearest example. Rather than one population being dark-skinned and another light-skinned with a sharp boundary between them, pigmentation changes gradually as you move across latitudes. This pattern closely tracks ultraviolet radiation rather than racial categories. The same logic applies to many other traits once used to define race. Frequencies of blood types, genetic markers, and certain skeletal characteristics do not cluster into neat racial packages. Instead, they form overlapping gradients shaped by migration, gene flow, and long-term population interaction. When anthropologists began mapping these traits across regions rather than forcing them into categories, the data told a very different story, one of continuity rather than division. Now, this part is important. Clinal variation explains why racial typologies repeatedly failed. If traits vary gradually across geography, then drawing firm lines between groups will always be arbitrary. Those boundaries cut across natural gradients and create artificial divisions that do not reflect biological reality. Early typologists weren't necessarily careless. They were working without population-level data. Once that data became available, the clinal nature of human variation became impossible to ignore. What clines ultimately show us is that human diversity is real, patterned, and meaningful, but not in the way race theories assumed. Variation reflects adaptation, movement, and shared ancestry across space, not discrete biological races, and this realization fundamentally reshaped how biological anthropology understands human difference. Let's revisit what we've covered in this section. We traced the origins of racial thinking and saw how early scholars, working with limited tools and strong cultural biases, tried to divide humanity into fixed categories. Then we followed the scientific challenges that dismantled those assumptions, Boas showing how environment shapes traits once thought to be rigid, Du Bois revealing the social roots of health differences, and genetic studies demonstrating that most variation exists within populations rather than between them. Finally, we looked at clines to understand why human diversity doesn't fall into neat boxes, but instead forms continuous, overlapping patterns shaped by geography, migration, and lived experience. Now, this part is important. None of this means humans lack biological variation. Quite the opposite. Humans vary enormously, but that variation doesn't align with the racial categories created centuries ago. When we use race as a biological explanation, we end up misunderstanding both the science and the people. But when we use population, environment, and adaptation, we gain a clearer picture of how and why humans differ. You might wonder why this matters beyond the academic setting. It matters because ideas about race still influence how societies interpret difference. When we understand that human variation is clinal, flexible, and responsive to environment, we become better equipped to question assumptions about inequality, identity, and ancestry. So as we move to the next sections of this module, keep in mind that human differences are real, but the lines people once drew to explain them were not. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.